Hi everyone, thank you for joining. I'm Emilien Pousse. I'm a postdoc working at the Northeast Fisheries Science Center in Mitford, Connecticut in the US. And today I'm going to present my work where we are projecting the effect of ocean acidification on warming and two Atlantic surfclam population by using a dynamic energy budget model. So you all know the story, carbon dioxide emissions are rising in the atmosphere. A quarter is taken up by the oceans and that leads to decrease the pH of the water. So this ocean acidification has a lot of effect on bivalves. So here is a review from GASO in 2013, and they summarize all the different kind of effect that OA can have on bivalves. So we know that bivalves bioenergetic is affected by OA, mainly by an increase in maintenance costs and a decrease in feeding, and that leads to observe some delay in growth rates. So in this presentation, we're going to try to understand how OA affects bivalves' energy budget. And by using modeling, we're going to investigate what could be the consequences in the future for fisheries and aquaculture. Okay, so the aim of this study is to calibrate a DEB model in which we will add the PCO2 concentration as a third forcing variable. And we will test the hypothesis that PCO2 affects ingestion and maintenance costs by using experimental measurements. And then, by using this model, we will make some projections to investigate the future effect of acidification and warming on different bivalve population by implementing different scenarios. The species we choose to work with is the Atlantic surfclam, Spisula solidissima. That's a really important bivalve for the Northeast fisheries. That's one of the top bivalve produced in this area. We know that these species live from subtidal areas to around 60-meter depth. We also know that population in some places are already moving offshore to avoid high temperatures inshore. So we know that temperature highest to 25 degrees C will negatively affect their growth. And the two offshore areas we are studying are the Mid-Atlantic Bight. That's the place where inshore population are already moving to, and Georges Bank, that is a hotspot for bivalves. So to calibrate this model, we ran a 12-week experiment where we exposed 560 individuals to 3 different PCO2 concentration. And every 2 weeks, we took those animals, we measured their feeding, respiration, excretion, and their growth. So concerning the effect we observed on feeding, here you have the standardized clearance rate as a function of PCO2 concentration for our 3 different PCO2 treatment. So as you can see, there is a significant effect on clearance rate, with clearance rate decreasing with increasing PCO2 concentration, and there is no threshold effect. And by comparing those results with cilia bit frequencies versus PCO2 concentration, as here in this graph from MESEC in 2020, so we can see there is a negative and linear effect from PCO2 concentration on cilia bit frequency. And actually, cilia bit are really important, and they are linked to feeding, because when cilia are moving, they are making a water current that will go through the gills of the bivalves, and that's how they can filter the water and select the particles in the water. So that's linked to feeding. And then to integrate this effect in the DEB model, we introduced a coefficient that will allow us to scale the ingestion flux as a function of the PCO2 concentration. So we estimated the value for this coefficient for the medium and the high treatment as the ratio between standardized clearance rate relatively to the control. So we have those points. We were able to calculate the slope and the boundaries. So below a concentration of 1011 microatmosphere, the coefficient was equal to 1. So there was no effect on feeding. And above 6,778 microatmosphere, the coefficient was equal to 0. So no more feeding. And then between those boundaries, we calculated the value for this coefficient with the equation that is written just here. And so we just needed 2 parameters to estimate this coefficient as a function of the PCO2 concentration. And then considering the effect we observe for the maintenance costs, so respiration and excretion, here you have the energy loss through respiration and excretion in joule per day as a function of the PCO2 concentration. So as you can see, there was a significant effect, but the relationship looks like an inverse U-shape response. So there is a threshold effect. And when we compare this with other results published, so for example, here with muscle, you can see that this is something already described. And actually, this response comes from a switch in the nitrogen metabolism. So when a PCO2 concentration threshold is reached, some animals are able to switch into to enter into a metabolic depression, and that will change how they use their energy. They will use different substrate. They will use nitrogen to buffer the extracellular pH. And that way they will be able to save some energy. So that's, we're guessing that is what happened for our surfclams here. So to integrate those maintenance overcosts in the DEB model, we did something similar to what we did with the feeding. So we assume that there will be maintenance overcosts increasing linearly until a threshold is reached. And when this threshold is reached, then there's no more overcosts, it remains steady. So same thing that with feeding, we needed two concentration boundaries, a low and a high. We assumed that the lower PCO2 concentration boundary would be between 566 and 1380 microatmosphere. That's the low and the medium PCO2 treatment. We assume that those overcosts were linked to surface, and then with the equation that you can see here, we were able to calculate those maintenance overcosts that were added to the regular maintenance costs and also corrected with the temperature. And then we calibrated the model and the maintenance overcost on dry flesh mass evolution as a function of the time for the three different treatments. So as you can see here on this graph, in black, you have the surfclam that are exposed to the low treatment, in orange, the medium, and in red, the high. So we observe a significant effect on the growth with delayed growth when PCO2 concentration are increasing. So the picture on the right shows the different sizes at the end of the experiment. So you can see that there's an important effect. Here you have the standard model, so without any effects. So that's the control condition. On top, this is the dry flesh mass, and on the bottom, the shell length. So no PCO2 effect. You can see that the model fits well with the measurements. And then we calibrated the model for the intermediate concentration and the high PCO2 concentration. So the medium treatment is in orange on the left, and the red corresponds to the high PCO2 concentration. So here too, you can see that the model fits pretty well with the observations. So to validate our model, we simulated the growth of a 1 centimeter surfclam over 25 years with forcing variables going from 1994 to 2019. So we downloaded the food and the bottom temperatures from the Copernicus dataset that provides satellite data. And we obtained the PCO2 concentration from the ocean soda datasets from NOAA. We compared the growth with observations from the NEFSC clam survey, where they sampled clams in Mid-Atlantic Bight and the Georges Bank areas since 1982. And here, as you can see, the models fit well with the observation. So we have a model that is validated. So now that our model is validated, we are able to make some scenarios. So we base our approach on the different IPCC scenarios to obtain the PCO2 concentration increase, and RUM's model to obtain the temperature increase in both areas, Georges Bank and the Mid-Atlantic Bight. So from that and from the variability we had in the 2019 year, we applied those rates, and we are able to obtain a daily variability for the next 100 years. We also made the hypothesis that food will remain identical to what we obtained from 1994 to 2019. And with those environmental conditions, we are able to use our DEB model to make projections in 2 areas following 4 different RCP scenarios over 100 years that we divided into 4 25 years period going from 2020 to 2120. Okay, so here you have the shell length projections for the 2020 to 2045 period for Georges Bank on the left and the Mid-Atlantic Bight on the right for the RCP 2.6, 4.5, 6, and 8.5. Actually, you can see almost only one line because for this period, there's no effect. All the different RCP scenarios induced almost the same growth. So for the 2045 to the 2070 period, you can see that there is a small positive effect for the youngest stages, a positive effect due to ocean warming. But if we take a look at the final length, we can see that it's almost the same. For the 2070 to 2095 period, we can see something similar with the beneficial effect of warming is higher, both for Georges Bank and the Mid-Atlantic Bight. But that's the first moment when OA will have a negative effect that is observable for the Mid-Atlantic Bight. So if you take a look at the right, you will see that there's a few millimeter differences between the RCP 8.5 and the RCP 2.6 for the around age 20 to 25. And for the last period, 2095 to 2120, we can see that there's still a beneficial effect from ocean warming, but at the end, for the surfclam exposed to the RCP 8.5 conditions, the final length will be lower than the other scenarios. For example, for Georges Bank, there will be a 0.5 centimeter difference, and it will be 1.3 centimeter difference for Mid-Atlantic Bight. Also, if we take a look at the moment where we observe the first negative effects, they will appear around 2107 for both areas. We also did some measurements to assess what would be the effect on reproduction and maturation. So here you have the age at puberty for the 4 different RCP scenarios, and still for the 4 different periods. We just showed the result for Georges Bank here. And so you can see that with RCP 2.6, the age at puberty will remain the same. But for all the other scenarios, it will come early. And if you take a look at RCP 8.5, the decrease is really important with something like more than 5 months difference. And we also calculated what would be the mean energy allocated to reproduction per year for those 4 RCP scenarios and those 4 periods. And we saw something interesting for RCP 8.5. We can see that a peak will be reached during the 2070 to 2095 period. And after, for the 2095 to 2120 period, it will start to decrease because of OA. So that could lead to important effect on the fitness of this species. All right, so the take-home message of this talk was that PCO2 affects maintenance costs and feeding. The first effects we observed on growth were around 2100 if RCP 8.5 is realized. We also saw that temperature will provide a small advantage for young individuals, but at the end and regarding the effect on reproduction, population could be threatened before 2100. The further work we would like to do now is to study the effect of extreme events, implement different scenarios with temperature or PCO2 concentration peaks, develop also the scale effect for oysters and calibrate the same model for sea scallops in order to make some comparisons. And there's also a need for more experimental data to investigate a possible cross effect between the temperature and the PCO2 concentration. And finally, I just would like to thank all the people that helped me during this postdoc. I'm now at the end of almost 3 years postdoc, so I'm really thankful for everything. And I would like to thank you for your attention.